Hello, my name is Sridevi. I'm from Ernakulam. I live here with my family. I completed graduation and post-graduation in biotechnology, and I worked as a biotechnologist. Later, I worked as a medical coder in some MNCs, and in 2017, I started my career as a full-time home tutor. Then in 2022-23, that time I worked as a medical coding tutor in a private institute, and in 2024 again I continued my career as full-time home tutor, and in 2025 I started my career as an online tutor. So I'm from a life science background, so I'm focusing on biology subject that includes EVS, botany and zoology. Okay, so today I'm going to take a class on Mendel's law of inheritance. Okay, Mendel's law of inheritance. So this chapter, this portion is from CBSE 10th standard biology, and Mendel's law of inheritance that we studied in high school session. That law of inheritance, that means now we are more advanced in genetic field, and now we doing gene knocking, recombinant DNA technology, we producing vaccines, we doing designing in genetics. So lot of variations or lot of advantages happened from the previous era. So because of Gregor John Mendel, he formulated these laws. Because of these laws, we came to aware about the inheritance, variation and genes, this kind of things. So now we are in an advanced mode. Okay, so first we should know about who was Gregor John Mendel. He was a botanist and he was a Christian monk, and because he formulated these all laws, he considered as the father of genetics. Okay, he considered as the father of genetics. So he conducted his experiments in pea plants. Okay, that is Pisum sativum. The scientific name is Pisum sativum. Normally in his monastery they planted the Pisum sativum, that pea plants. So he observed that time that every generation there is some character changes happens. That means sometimes they are long, in some generation they are short, their seed coat color is different, their flower color is different. So he think over it, why this kind of change is happening in every generation. So he was a botanist, he decided to invent the process that happening behind this phenomena. So he studied the inheritance and variation. So what is inheritance? What is the meaning of inheritance? Here you can see the word inheritance and variation. What is the meaning of inheritance and variation? Inheritance means the transfer of traits from the ancestors, from the parents to the offspring. Okay, variation from the word itself you can understand the meaning. That means differences. Okay, differences among the individuals. For example, if we consider a family, the whole members are not look like same. They have variations in their physical and mental characters. Okay, so that means variation. Okay, so he worked on this incidents, that means the inheritance and variation. See, so he formulated three laws: law of dominance, law of independent assortment, law of segregation. So first we are going to study about law of dominance. Okay, law of dominance. From the word dominance you can understand that means there is a dominant character. Okay, so what he did was that I included a picture here so you can understand it better. He took a tall plant, a pea plant, and a short plant, and he allowed them for pollination. Okay, so what happened? They did a mating here. So what happened is that in the first generation, the F1 generation, This is F1 generation, that means first filial generation, which produced all tall plants. Okay, these were all tall plants. And in the next step, he did cross pollination, not cross pollination, self pollination. Self pollination between the F1 generation, the first generation, you can see. So in the second generation, he got another result. So what was that actually? Here the tall plants, while they transferring the capital T, which representing tall, okay, tall character, and when the gamete formation happened, the tall character segregated from the tall plant, and from the short plant, what the small t, which represents shortness, that segregated through the gamete formation. So in F1 generation, you can see this is capital T and small t. That means it is the mixture of their parents. The parents were pure lines. You can see there is no mixture. There is no mixture happen. The mixing not happened there. But this is, you can see a mixing happened. So this is tall plant, but it beared a short gene also, a short allele also, but it not expressed there, because the tallness, it is the dominant character, and the dominant gene, this dominant gene masked this recessive gene. Okay, so to find out that dominance, he did a self pollination between the F1 generation. So what happened here in the F1 generation? What happened? There are two gametes will be there. Okay, so what happened? This is for tall, and this is also for tall, because there are dominant character is present. Tall, again this for dwarf. Okay, so he got a result that the genotypic expression was 1:2:1. 1:2:1. 1:2:1. 2 means these two. Okay, that why the genotypic expression is like this? Because this is a pure line, this is a pure line. Okay, but this is not pure, they are a mixture. Okay, they are mixtures. So that's why they considered a separate group. Okay, that is why 2 is the 1:2:1. That is the genotypic combination is 1:2:1. But the phenotypic ratio, you can see the phenotypic ratio 3:1. What is phenotypic ratio? What is phenotypic ratio? Phenotypic means the observable trait, the characters that we can see outside. So while we seeing this picture, you can see that they are tall, this is short. Okay, so normally we writing 3:1 because this is the observable trait. Okay, so from this findings, he concluded that in a pair of contrasting traits, one trait masks the effect of other. Okay, one trait, that means the dominant trait masks the other recessive trait. Okay, so this is applicable in every organism. Okay, so this is the law of dominance in a pair of contrasting characters. Contrasting characters. Okay, in the pair of contrasting means different, opposite characters. One trait masks the effect of other recessive trait. Okay, this is law of dominance. And here he observed that 3 by 4th of the F2 plants were tall and 1 by 4th were dwarf. Okay, that means new combinations or the variations are happening when we consider the parent plants. Okay, they are totally different from the parent plants in genotypically. Okay, and again he concluded some things, some points, like Mendel concluded that traits are passed down as discrete units. They are transferring, the traits are transferring as discrete units. Okay, discrete units which he called factors. Okay, which he called factors, and now known as genes. Now we calling this discrete units or this genetic factors, what we calling right now, genes, the term which was coined by Wilhelm Johansen. Okay, Wilhelm Johansen coined the term gene. Okay, the tall trait was dominant and the dwarf trait was recessive. That means it is expressed in F1 generation. The dominant were expressed in F1 generation, and that hidden in F1 generation. What was hidden in F1 generation? What character? Shortness. That is reappeared in F2 generation. That you can see in this picture. It was hidden in F1 generation, the shortness, and it reappeared in F2 generation. Okay, these were the findings he did, and here we should know the term meanings. Alleles. Okay, allele is, what is allele actually? Each individual has two factors for a trait. Okay, for example, tallness is a trait, is a character. For that, two factors are there, this and this are the two factors. Okay, one from each parent, it is transferring from each parent, that you can see in the picture. Okay, this tallness, this allele transferred from the tall parent, and this allele is transferred from the short parent. Okay, and they come together to produce a new gene combination. Okay, so each individual has two factors for a trait, one from each parent. These are called alleles. This is one allele and this is another allele. Okay, this is one allele, the capital T is one allele and the small t is another allele. So this is I want to say about alleles, and also I share one picture. Yeah, in this picture also you can see this is a chromosome. During the gamete formation, it separates into alleles. Okay, this also relatable to you. This how this gamete formation help to separate the alleles. So this separate out, the alleles separate out and move towards the next generation. Okay, and genotype and phenotype, that I already mentioned. Genotype is the genetic makeup. Okay, genotype is the genetic makeup and phenotype is the observable trait. Okay, next is Punnett square. Normally we drawing some square like this. Okay, sorry. And this one. Okay, when we drawing like this, normally what we get, if we cross these two, normally we get capital T here, and when cross these two characters, what happens? When they join together, what happened? Again capital T, small t. That means it's for tallness. Again we cross this two characters, what happens here? Again we getting tall plant, and while we crossing small t, what happens when they unite? Short plant will produce. So we drawing this kind of diagrammatic representation. Normally we calling this representations Punnett square. It is very easy to understand the offspring ratios. Okay, this is about law of dominance. Next is law of segregation. Okay, law of segregation, that means law of purity of gametes. Here the alleles of genes separate during the gamete formation, that we already know. Each gamete carries only one allele for a trait. That also we discussed right now. Each gamete carries only one character. Each gamete, that means this is a it's for this is for tall plant. Okay, in F1 generation, and here two gametes will form. Okay, so each gamete carries only one allele for a trait. During the fertilization, the gamete combine randomly, leading to new allele combination in offspring. Okay, that means I will write it. This is for tall, so normally this gamete will form here, and this is for short, and short this gamete will form here, and during the fertilization what happens? A new gene combination were formed here. Okay, so this is, he says that each gamete carries only one allele for a trait. For a character there is only one allele is there, and during the fertilization the gamete combine randomly. These gametes combine randomly to produce new allele combinations or new gene combinations. Okay, new gene combinations in the offspring. This is also tall and this is also tall, but the difference is that, what is the difference? It have another gene which is for shortness. Okay, so a new variety were formed. For example, a heterozygous plant produce gametes with either T or small t, never both. That means this is a heterozygous. What is heterozygous and homozygous? That I will. Okay, here you can see only one type of alleles are here. This is for homozygous. Homo means same. Okay, homozygous. And this is heterozygous. Why it is heterozygous? There are two different alleles for a character. Okay, two different alleles for a character or a trait. Okay, this is for tallness and this is for shortness. Both represent what? Length. Okay, so here two type of gametes will form. Okay, but they will not transfer as pair. It never, the alleles transfer as pairs. They will transfer as in the single form. Okay, only one allele is transferring from parents for during the gamete formation. Okay, so law of segregation states that a heterozygous plant produces gametes with either capital T or small t, never both. Both alleles never come together during the gamete formation. Only one gamete means only one allele coming from two type of parents. That means the capital T and the small t from two parents. Okay, it will not come together. during the gamete formation. And here the law of segregation states that the two copies of each genetic factor segregate during the development of gametes. Okay, two copies of each genetic factor segregate during the development of gametes to ensure that each parent offspring attain one factor. Okay, that we already seen here in this crossing also we have seen this. Okay, so the two copies of each genetic factor segregate during the development of gametes to ensure that each parent's offspring attain one factor. Only one factor is attaining from each parent, from the paternal side and from the maternal side. Okay. Next, the law of independent assortment. The law of independent assortment. Here, he did a dihybrid cross. Okay, here he did a dihybrid cross, and here he proved that alleles for different traits like seed color and seed shape, this is two different characters, seed color and seed shape, they separate independently during the gamete formation. Okay, gamete means the sperm or egg. So during the gamete formation, the different traits alleles separate independently without interfering each other. They not interfere each other. Okay, so to prove this, what he did was he crossed yellow round plant with green wrinkled plant. Okay, green wrinkled plant. So the representation will be like this: yellow round. Okay, yellow round and green wrinkled. Okay, like this. So in the F1 generation, the offspring are yellow round, because here you can see the yellow round is the dominant character and the green wrinkled. Okay, green wrinkled is the recessive character. So normally during the first test cross, what happened? The first generation, the F1 generation, this is F1 generation. Okay, the first filial generation gave yellow round plants. Plants means the seed color. Here you can see the seed color. Mendel cross seed plant with yellow round and green wrinkled. Okay, this is seed plant of seed. Okay, so here yellow round is the result in F1 generation. So he did a cross pollination between the F1 generation. So what happens normally? This is F1 generation. So what are the possibilities of gametes here? Yellow round, yellow wrinkled, green round, green wrinkled. Okay, so we this is the example for Punnett square. Okay, this is also this is the example for Punnett square, and here you can see yellow round, green round, yellow wrinkled, green wrinkled. The same gametes we wrote on this side also. Okay, the gametes from heterozygous parent, and here also the gametes from heterozygous parent. Okay, when we cross this, means when these genes come together, what happens? When these genes come together, all were yellow round. When this comes together, you can see there is a green gene is there, but it is masked by the yellow. So normally what happens? Yellow round is the result. And in the next also you can see there is a gene for wrinkled, but the result is yellow round. Why it is? Because the round is the dominant character masked by the wrinkled. And again you can see when these two type of genes come together, it again yellow round, because the dominant characters masking the recessive genes. Okay, so we did next cross. Then what happens here? Green round and yellow round. So normally this recessive genes were masked. It's become again, the result will like the same, yellow round. And when did this type of cross, what happened? Here you can see small y and small y. That means green, green, green. So these genes come together. What happens? Green round. This is a new combination. Green round, which is totally different from the parent gene combination. A new combination were formed. That was green round. And again these two crosses happens. What? Yellow round. Again it is yellow round. And these two, green round and green wrinkled. Here again new gene formation happened, because there is recessive genes are came together, same like in this case the recessive genes came together, so they expressed as dominant. So it is green round, a new combination formed. And we consider the next row. What happens? Yellow round. Okay, this is again yellow round, and in this case this is also yellow round. And in this case this again yellow, but there are small r and small r here. Here is a small r and here is a small r. Small r for what? It is for wrinkled. So yellow wrinkled. This is also a new combination from the, which is different from the parent genotype. And again you can see yellow wrinkled cross with green wrinkled. Again the wrinkled, the recessive genes came together. So what happened? Yellow wrinkled. Okay, yellow wrinkled. This is again a new gene combination. Okay, and the last one, green wrinkled crossed with this one. Definitely the outcome will be yellow round. Okay, next these both. Here small y, small y, small y for green. So when they came together it become dominant. So green round, green round. Okay, green round. There is a capital R, it is for round. So green round, it is a new combination. Okay, then we crossing these two. Here yellow is a dominant color and small r are there, are coming together, so it become dominant. So what happens? Yellow wrinkled. Okay, new combination happened. And again here green wrinkled, green wrinkled, pure lines are there. Okay, so what happens? Same like the parent plant. Okay, green wrinkled. So in this combination you can see 9 plants were yellow round and 3 plants were, you can see these 3 plants were green round, and again you can see these plants. What are they? Yellow wrinkled, yellow wrinkled, and only one is there for green wrinkled. Okay, so the pure lines in case of dominant is 9, and in case of shortness, means in case of recessiveness, not shortness, in case of recessiveness is only 1. Okay, so 9:3:3:1 is the ratio that we got here. Okay, that is we got here, and he concluded that this ratio shows that seed color and seed shape are inherited independently. Okay, it is not mixing each other. The seed color and the seed shape are inheriting independently, creating new combinations. You have seen new combinations here, creating new combinations. That means yellow wrinkled, green round, that were not present in the parent plants. Okay, new combinations were happened. That means inheritance and variation both happened here. Okay, so this is the conclusion he did, and he formulated the law that alleles for different traits separate independently during the gamete formation. Alleles for different genes sort into gametes independently of each other. That means different characters have different genes, they are sorting out during the gamete formation, but they not interfere to each other. They are sorting out, they are transferring independently. Okay, example, inheriting a gene for seed color doesn't affect the inheritance of gene for seed shape. Okay, this is the finding. This is the finding he concluded after the dihybrid cross. Okay, so normally asking in question paper, what is the ratio of dihybrid cross is 9:3:3:1 ratio. Okay, so the ratio will be like this: 9 pure lines in case of dominance and 1 in case of recessive character, and the remainings are new combinations. Okay, so we now talked about 3 laws: law of dominance, law of independent assortment, law of segregation. Here why we need this kind of why we need to understand these laws, because it explains the genetic diversity, how it arises. Okay, how the genetic diversity arises and the shuffle of traits, okay, during the sexual reproduction. And the fundamental concept of genetics. We have some, because of these laws, we have some concepts. Based on this concept, we formulated other theories and we did some practicals also. Okay, it is fundamental concept in genetics showing how gene located on different chromosomes assort randomly. Okay, normally in chromosomes the genes are located in very different locations, but during the formation, during the gamete formation, they assort randomly. Okay, they assort randomly. And what is the importance of these laws in genetics? Okay, Mendel laid some basics, some foundation to the modern genetics. Based on these laws, we can at least understand the pattern of inheritance, the pattern of variation. Okay, and these principles are crucial for understanding the chromosomal theory of inheritance, the linkage, mutation, and genetic disorders. Okay, if we cannot understand this inheritance and variation, we cannot understand the location of genes, and also we cannot understand the genetic disorders. And this law of segregation, law of independent assortment, and law of dominance, these all laws are very beneficial to create new breeds. It is very useful in the field of agriculture. It is useful in the field of cancer biology, in the field of recombinant DNA technology, cloning, and genetic mutation. Genetic mutation, and also gene knocking. That means we can delete some genes, and same like that we can add some genes. That is very beneficial in case of antibiotic production. Okay, so vaccination, in case of vaccine production, the gene mutation and gene knocking and addition of genes, these all things are very important. Okay, now we applying these all procedures in modern genetics. So we studied about law of dominance, that is one allele masks another, and the law of segregation, that is alleles for a gene separate during the gamete formation, and law of independent assortment, that means different gene pairs separate independently. Okay, so these are the basic concepts of law of inheritance. Okay, so it is very beneficial to understand the linkage, genetic mutations, inheritance. These are kind of chromosomal phenomena. Okay, and because of this now we have gene editing, that I already discussed right now, CRISPR. Okay, this is a new technique for gene editing. Gene editing is very crucial in case of for the designing babies. For example, some parents have hemophilia. They cannot, if they have a progeny, means if they have a child, definitely that child will carry the same character, means that same disease. And in case of cancer also, lethal kind of cancer, that parents have cancer, both the parents have the same cancer condition, definitely the child will have that cancer condition. So in that cases we can edit the gene, we can delete the gene during the small stage of embryo development. We can delete the gene, and it's very helpful for producing new breeds, genetically modified crops, genetically modified animals. Okay, so the law of inheritance have lot of importance. So this is about this chapter. So thank you for your time. Have a nice day. Bye. Thank you.